With COVID-19 this year has come a new zest in the research around drug and drug delivery technology. One company that specializes in looking at improving the safety and effectiveness of drug delivery systems is N4 Pharma. Nigel Theobold is the chief executive. He joins us now from Derbyshire. Nigel, it's good to talk to you. Let me just ask you first of all, I've explained very briefly about what it is that N4 does. Give us some more depth of detail to give us a better idea.
Okay, well, N4, we're a preclinical pharma company, and we specialize in a unique silica nanoparticle that is a delivery system for vaccines and cancer treatments. So we've got an exclusive global license for this technology from the University of Queensland in Australia. And what it is, it's a tiny little nanoparticle that you can load nucleic acids onto, so think DNA, messenger RNA, and then deliver them into the cell so that it can carry the payload into the cell so it can do its job and produce the antibodies required for cancer treatments and things like vaccines.
Give us more details about precisely the areas you're involved in, because as I said at the top, in the context of COVID, everybody is now more interested in this sort of area of technology. Just give us some more details. You referenced cancer particularly, but does this expand out into other areas?
Exactly, it does interact. So we're really involved in the delivery of nucleic acids, and that's the key thing. So as I mentioned, things like DNA and RNA and messenger RNA, which you use for cancer. And we're looking at three key areas. The first is injectable vaccines, and the other area is the potential oral delivery of vaccines. And then finally, it's the intratumoral or direct treatments of tumors in terms of cancer therapy. And the way what we have is this unique particle. It's a bit of a spiky particle. It's hard to sort of describe. Well, I often describe, if you've been traveling in the Far East, you'll know something called a rambutan fruit, and it looks like that. We in the UK, we tend to get it. So if you go walking, you tend to get these little burrs stuck on your socks. It's a really bizarre analogy, but they're spiky, tiny little hollow centers with these spikes on. And those spikes are what makes our delivery system so unique, because those spikes trap DNA and RNA. Because DNA is like a double big loopy structure, and it's hard to get into a lot of delivery systems. And what that does is it allows us to protect the nucleic acid and ultimately deliver it into the cell. And we use it as an alternative delivery system for nucleic acids. I've got a really good video to show you exactly how the particle gets into the cell. So if I show you that now, what you'll see on this video is that the elements in red are the Nuvex system that's gone into the cell. So it shows you how it's taken up into the cell. And it shows you, this is a time course. So you show by about 12, 13 hours, we've got this good green signal coming, which is the production of protein within the cell. And as it progresses, we get more and more green coming through. And again, what's important here is that the cell is still growing. It's still very healthy, so there's no toxicity to the cell. So the Nuvex has got in, it's delivering the protein, and this protein will ultimately be used to deliver the peptide that creates the antibodies for vaccines and cancer treatments. So that's just a little video that shows exactly how our system works and why we're focusing on the same nucleic acid delivery.
And you can replicate this across a number of different areas, can you? I mean, we're talking there specifically about one area you're concentrating on at the moment. But if a pharmaceutical company came to you and said, can you replicate your process using, for example, a vaccine for COVID-19? I talk about it in this context because it's what everybody's knowing about at the moment. But,
exactly,
but it is a possibility for you just to do that.
Absolutely, for any vaccine. So what's happening recently is that mRNA particular, but nucleic acids are now becoming very much at the forefront of vaccine technology. But it's not just for COVID. COVID's given the opportunity to show how it works, and they're working very well. But you can use nucleic acid for any vaccine: influenza, hepatitis, dengue fever. And as I say, you can also use it to deliver a special type of nucleic acid called an siRNA, which a lot of people do for cancer treatments. So the number of applications are immense for our system.
Now you've just raised, I think, what, £2 million and a placing at 8
Yes.
pence a share. What's the reason for that? What are your intentions with the money?
Sort of hopping back to, the good news that Pfizer have delivered this week with vaccines has really brought mRNA right to the forefront. So mRNA is a type of nucleic acid. We've done a lot of work on DNA and some work on mRNA. What we will be doing with the new funds is accelerating and extending the work that we have been doing on mRNA so that we can show, for example, the recent data that we've shown how we can load, store, and keep vaccine in an ambient temperature. For DNA, we want to do that work for messenger RNA to make it really applicable to those vaccines that are right at the front of this current approach. We'll also be doing our most comprehensive in vivo study using an optimized version of our NUVEC system. So all the data that we've been generating over the last 12 months on how to use NUVEC, we'll be putting into the biggest study that we've done yet, which again is proving with COVID and other things exactly how NUVEC behaves. And also increase our business. We're doing this now in an environment where vaccines are becoming extremely interesting for the pharma community. So to increase our BD efforts, business development efforts, and also we're looking at complementary technologies that we can add to our portfolio alongside NUVEC.
Okay, let me bring up a share price chart just to get a better idea of the context of what's been happening. This goes back to the back end of 2019, so you can see what's been happening this year. And you can see quite clearly from the lows that we had back at, 2.1 pence back at the beginning of March to the highs, you have, as a company, benefited from the money that's been going into this sector, clearly. Since then, there's been this little bit of a pullback. At 8.2 pence, which is where we are at the moment, what's been priced in, bearing in mind you've had this 2 million raising at 8 pence a share, and you've got a market cap, I think, of something like around about 13 million at the moment.
Yep.
So what are investors looking at 8 pence a share?
Well, they're looking at a company with a huge amount of potential, in my view, because that sort of valuation around where we're at. So we're at the start of our journey, really. We've got this delivery system, we're showing the proof of concept data, and what we need to do is to start partnering with major players, because we don't own the DNA or the RNA. The DNA or the RNA is the thing that will be owned by the likes of Pfizer or Moderna, and we need to start working with them. They will be licensing our system to bring new vaccines to market. And well, you look at the size of the opportunity and the sort of numbers that are involved in this market space, there's a big potential to play, and a new delivery system in this space I think is going to be really exciting. So we're very much at the start of our journey.
I was going to say, how far down the path are you to that? Explain a little bit more about what you've got to do before you start engaging with these pharmaceutical companies that ultimately could end up using your product.
So yeah, we've done a lot of work showing how NUVEC works. I've just shown you the video to see how we see how it works in cells. We've done a lot of research looking at standard plasmids of DNA called albumin. We are now using the COVID work as even further proof of concept with a real product. So we know it works. We've shown people it works. We show it can deliver the antibody. But what we're looking at is trying to work out what's our real point of difference. And one of the things that's been really interesting that's come out of the COVID work is what I call the difference between efficacy and efficiency in a vaccine. So the efficacy of a vaccine is how well does it work, right? And you've seen the numbers that have come out from Pfizer and Moderna recently. They're getting 95% efficacy, which is very, way beyond the expectations. So let's be realistic. Nobody's going to beat that. No one's going to beat 95%, and no one should be trying. If you get 96, 95, well, not a lot of difference. But the real test will be the efficiency of the vaccine, and that is how well and how easy it is to roll out. And the one problem that these vaccines, that nucleic acids, especially mRNA and DNA as well, because DNA needs electric equipment to make them work currently, is the fact that you need to store the vaccine at minus 70 for Pfizer and minus 20 for Moderna. So there's a huge issue with rolling it out. So you have this efficacy from the vaccine, but you then need to have it rolling out. Now, we've shown through our work that we can store it, freeze dry it, store it at fridge temperature for DNA. We're looking to do that, as mentioned, with the new funds for RNA. But that gives us a real point of difference, because if people have struggled to get DNA and RNA into systems and then store them effectively, and we can do that, that gives us a very strong point of difference in the market. So that's really what, where we're looking at. How do we, because the efficacy will come from the RNA or the DNA itself, not from us. What we've got to do is show we can get it in, and then show we can get it in a really easy to use way.
And any competitors in this market particularly?
Oh, it's a massive market. I mean, our competitors are the systems that exist, so lipid nanosystems. There's competitors that have electroporation system, viral vectors. There's a number of different ways that you can get vaccines into the cell. There are different antigens that are placed into vaccines. But as I say, there are very few that actually work well. For DNA, there's very few will work well for, and you've seen from our video, it's a video of a plasmid DNA that gets into the cell, that transfects within the cell, and there's no electroporation machine in sight to make that happen. So we're pretty confident that we've got a very strong delivery system, and that we will be able to compete with a lot of people in the marketplace.
Yeah, look, Nigel, good luck with everything, because it sounds a fascinating area, and of course something that humankind needs in order to improve, as you said, the efficacy, the efficiency in drug delivery and the safety and so forth. So all power to N4's elbow. It's a pleasure to talk to you. Thanks indeed for your time today in catching up with us on what N4 Pharma is doing in this area. That's Nigel Theobold. He's the Chief Executive.